So in recent years, there's been quite a revolution in biology because they have more and more sophisticated instrumentation. So they have microscopes that can really acquire images in 3D. And so biologists have contacted us because they need to outline structures in those images like cell nuclei. And so therefore in the lab we developed software that can help them to do that much faster in a much more reproducible fashion.
We developed a software that allows the biologists to quantify some things of their cells like size, intensity. It was something that before it was done manually, and it took really a lot of time, especially when dealing in 3D data, and now it can be analyzed automatically in a regular computer or even a tablet. What our software provides is different visualizations of the image data that the biologists have, and from there they have to just choose an initial position from our program to start. It appears some kind of a sphere, and from there they just have to drag it with the mouse close to the cells they want to analyze and click a button, and the sphere automatically adjusts the cell. 2D version of the plugin already works in tablets, so we have been experimenting a lot with it, and we'll in the future port as well the 3D version to these kind of devices.
So it was very important for us to put it on the web, make it available to the community so that as many people as possible can use it. And also, this is a very interesting research question, a challenging problem that required the development of rather sophisticated mathematical tools.